estamos grabando. Ok, muy buenas tardes a todos y todas. Vamos a iniciar un esfuerzo que creo que hemos hablado durante mucho tiempo en la comunidad de hidrología isotópica y de hidrología y de ecohidrología y de ciencias atmosféricas de Latinoamérica, que es tener un espacio regional que nos permita intercambiar experiencias, compartir ideas, apoyarnos con otros investigadores o investigadoras, de tal manera que podamos construir una comunidad más unida, intercambiar también tal vez pasantías, posibilidades de análisis, etcétera. Todo lo que pueda ayudar a incrementar la visualización principalmente. La calidad del trabajo es excelente, pero quizás algunas veces la visualización del trabajo nos ha faltado un poco. Entonces, la idea es unir al Caribe, la idea es unir a la región de Mesoamérica, la idea es unir también a la región de Sudamérica, incluyendo evidentemente que a los compañeros de Brasil, que creo que tenemos a Rafaela por aquí hoy. En principio vamos a tener un seminario cada 2 meses. Yo he estado invitando personas, ya tenemos algunas personas en la lista ya de espera para los próximos meses y vamos a darle oportunidad a personas del Caribe para que puedan hacer sus seminarios también en inglés. Vamos a darle oportunidad a estudiantes de maestría, doctorado o postdocs, especialmente los estudiantes de maestría y doctorado que quieran presentar algún trabajo, quieran prepararse para alguna defensa, quieran contar con un poco de experiencia y sabemos que existen muchas limitaciones para poder participar en grandes conferencias en Europa o en Estados Unidos, cuestiones de visa, cuestiones de financiamiento que hacen cada vez más difícil participar en esas conferencias. Inclusive para los que estamos acá, ir a esas conferencias requiere por lo menos de $3,500 por persona. Así que cada vez es más costoso y eso se ha ido evidenciando en muchas discusiones que hemos tenido. Entonces la idea es crear ese espacio, un espacio amigable. Aquí nadie viene a juzgar a nadie, queremos todos y todas tratar de mejorar la investigación y la visualización de nuestro trabajo. Entonces estamos como parte de eso tratando de volver a generar una edición especial. No hemos seleccionado todavía el journal, eso todavía tengo que comenzar a conversar con algunos editores, pero para ello tenemos que darles una propuesta bastante clara de la cantidad de papers. Generalmente si recibes unos 30 abstracts, si tienes suerte, tal vez la mitad terminan siendo artículos, así que se reduce sustancialmente a la hora de la publicación, eso hay que tomarlo en cuenta y generalmente un buen número para tener un special issue es posiblemente entre 12, 15 o 18 artículos. Vamos a trabajar en ello. La idea es trabajar en hidrología urbana, pero creo que podemos extender el tema a zonas tal vez periurbanas o dependiendo del estudio a otros dominios. Entonces, esa es la idea. Vamos a ver cómo nos va y trataremos de que sea una iniciativa que se consolide con el tiempo. Ahora, vamos a ver el teléfono. Ya, listo. Ahora vamos a presentar a nuestro primer invitado. Yo conozco a Cristian desde hace, creo que 12 años. Nos conocimos, creo que en AGU. Él estaba con un póster y yo estaba con un póster a la par y los ambos nos dimos cuenta de que estábamos hablando de temas similares o de Costa Rica. Y de allí hemos comenzado a trabajar desde hace 12 años. Ha sido una excelente oportunidad y creo que Cristian es una de las personas más capacitadas para poder hablar sobre el tema del uso de trazadores y modelación. Así que Cristian es un profesor de hidrología del Departamento de Geografía de la Universidad de Costa Rica y es el director del Observatorio de Agua y Cambio Global y del Sistema de Estudios Graduados del Departamento de Geografía. Cristian es profesor honorario afiliado al Centro de Investigación Tropical en Agricultura y Educación, más conocido como CATI, en Turrialba, Costa Rica, y también recientemente fue investigador invitado en el Leibniz Institute of Freshwater Ecology and Inland Fisheries en Berlín, Alemania. Su investigación en modelaje, en entendimiento de procesos hidrológicos para la generación de escorrentía, procesos biogeoquímicos, equidología, ha generado más de 100 artículos científicos y su índice de publicación, el índice que llamamos el índice H, es de 36, es altísimo. Su investigación trata de entender, como mencioné, ese funcionamiento a nivel tanto local como regional y global, e interacciones entre agua, energía, biodiversidad, ecología, seguridad hídrica, etc. Hoy nos va a tener un seminario que se titula El potencial que todavía no hemos explorado sobre el uso de modelación asistida por trazadores para mejorar el manejo ecosistémico y el manejo de recursos hídricos en diferentes ecosistemas. Tengo otra persona aquí invitada, listo. Así que, Cristian, muchísimas gracias por aceptar esta invitación y la audiencia es toda tuya.
Perfecto, Ricardo, muchísimas gracias por la introducción.
Cristian,
voy a compartir pantalla.
Sí, y creo que se han unido unas personas de idioma inglés del Caribe, así que creo que podemos ir con la idea principal de que era darlo en inglés para que ellos puedan atenderlo. Vamos a hacerlo de una manera lenta para que las personas de Latinoamérica también puedan entender. Creo que es importante incluirlos a ellos, de otra manera sería un poco difícil.
Okay, so yes, to be inclusive, so I'm switching to English, which is probably better for me, so you don't have to bear with my German accent in Spanish. Can everybody see my screen?
Yes.
Do you hear me? Okay, brilliant.
Yeah.
So Ricardo, thanks very much for the invitation to speak today. I also want to thank you for the organization of this Latin American and Caribbean network. I want to mention and thank you for your tireless generating isotope data, hydrology now beyond Central America and Caribbean. So to make isotopes more accessible, generating more data, publishing, speaking, making people aware of this potential we can find in isotope data. So big thank you for this, and I will start this seminar. Maybe acknowledging people. You only see my name on the first slide, but obviously there were a lot of people up and foremost, Ricardo, colleagues at University of Santa Maria Durán, with whom we both have collaborated quite a lot. Many other colleagues at the National University of Costa Rica. We have both the national colleagues studies in Costa Rica, and I will present a little bit of the type of modeling I'm doing, which incorporates stable isotopes. And obviously there is this goal of using the information we generate with models for water and ecosystem management. So we want to help decision making, and we want to provide data that is useful for management. And obviously the title, you can see that I think those type of isotope enabled models, they offer us a lot more that most people in hydrology or atmospheric sciences maybe are aware of. So I'm very keen on promoting the isotope-enabled models outside the isotope hydrology community. So this is why the title is called the unrealized potential of isotope-enabled models for water and ecosystem management. I will also not only talk about the advantages we find with this type of models, but also show some of the challenges and the limitations and maybe ways forward. So as a starting point, I want to show you this figure here, which is a little bit where are we at with our isotope hydrology and isotope-enabled hydrological models. So I will talk you through this. This is a simple Scopus keyword search for publications. What you see in red, this is the global land surface temperature anomaly.
Christian,
So obviously we have this.
Christian,
Yes.
maybe it's just me,
Yes.
but I don't see the slides moving.
Is it still on the title page?
I think so, yeah.
Maybe there's a delay. I switched to the next. That's a bit strange. Do you see this moving or not at all? So I'm back to the titles.
It's not moving on my end. Is other people having the same issue, or is just
Yes,
me?
I have the same problem. I don't see the slides moving.
Yeah.
Ooh, damn.
Yeah, it's not moving.
It's not moving at all. I don't know if I'm doing something wrong.
Maybe you can unshare and.
Let me just check.
Yeah. Now it's moving.
Do you see this?
Yeah.
Now it's moving?
Yes. I see the second slide. I see the slide with the graph and the publications.
Okay. So I didn't do anything. So maybe there is just a slight delay.
Yeah, at the beginning was kind of choppy. So I don't know if your internet signal is strong enough, maybe.
Okay, yeah, that might be. It's fluctuating as usual. So let me know. So if I'm talking and the slides are not moving, then please just interrupt and let me know. Okay. So what I want to show here is there are generally certain tendencies maybe since the 90s and 2000s. So the overall global land surface temperature is increasing. And then if we look at the number of published articles normalized to make comparable between, for example, climate and hydrology models. This is the gray line. Then we have this monotonous positive trend. Or articles that are concerned with any type of model uncertainty. And then we have a blue line that only starts in 2002. Those are papers concerned with climate change adaptation. And what we have here is in black, the overlying line. So those are any type of publications that in keywords or abstract or title mention isotope-enabled modeling. That can be climate models, that can be hydrological models. The thing is now that the absolute numbers for climate and hydrology models are 1,000 times higher compared to the isotope-enabled models. And the articles concerned with model uncertainty are 100 times higher compared to isotope-enabled climate and hydrology models. So there is we are lagging behind those other type of model approaches and model applications. The good news is that since 2007, you see this vertical line LAS in bold. This is when the first laser absorption spectroscopy instrument hit the market by two companies principally. So from there, moved from mass spec generated isotope data to simultaneous, at least for liquid water, deuterium and oxygen 18 measurements. So that kind of kick-started a lot of studies globally and also kick-started a lot of the publications we found globally. But there is this lag, so the isotope hydrology community is still lagging behind, and we to make our type of studies and data more accessible to the wider community, because there is this potential, and this is something I will be showing. So to switch slide here. It's working. Do you see the next slide?
Not yet.
No way. Ve.
Not yet.
No, parece que el Mac está teniendo problemas.
So I switched the
Maybe you
slide.
can
I didn't do anything.
turn off your camera as well, because maybe that's, I don't know, slowing your signal. But yeah, it's not moving yet.
It's not moving yet. Okay, just let me try this. I'm also pretty new to Teams.
Sorry about that. We can try Zoom next time.
No, that's all right. Been switching between platforms for the last 2 days, so just give me a second. This is the meeting, so I will switch off my camera to give it a little bit more control. Maybe back to. Do you see the slide now?
Not yet. Not on my end yet. Still the slide with publications and all of that.
Okay. I don't know. It should be there. I don't know what else to do, to be honest.
Maybe you can just go back and unshare your PowerPoint, and maybe not in the full mode, but in the different mode, we can actually see the slides moving. Other than that,
do you see the screen now?
Yeah, we see the screen. Another option is that you can share with me the presentation and I can pass the slides.
I mean, would this work? At least for most people, as it is.
Is it still like is like
yeah, it seems like it's frozen.
Yeah, I think your screen is not moving at all.
Okay. Let me just try to get this back up again.
Okay.
Sorry about that. How about now?
Maybe you can just have it like in that mode, and that will work. We are seeing the next slide now.
Yeah, no, now we see it.
So you see the slides and you know, the window with
Yeah, I mean we are seeing
you
are seeing the presentations. Can you move to slide 4, for example, just really quick?
I did just that. Did it change?
No, it's pretty slow. There is a huge delay, I guess. It is in slide number 3 now, but it's not moving.
Yes.
Maybe if you save it as a PDF and then share the PDF, that might work, because sometimes the PowerPoint is too heavy, so it doesn't move as fast as a PDF.
Okay, good point. I just export this as a PDF. Sorry about that. My internet should be fine. You hear me okay? So it's not, it's shaky. Just yes. Okay, I just will switch to PDF.
Okay, that should work. So in the meantime, for those who are just joining, I'm sharing in the chat the Excel file that is supposed to be a form to actually get contact information for you guys, and also to let you know about the special issue effort that we are doing. And if you really want to participate, there is some instructions on how to do it. So let me know if you have access to the chat and the Excel file, which is actually a OneDrive link. I can share it again because I think if people is joining late, they won't see the shot. Are you still there, Christian?
I am. I'm exporting.
Okay.
I don't know why it's so slow, to be honest. I mean, the PowerPoint is quite heavy, but nothing extraordinary, to be honest.
It might be one of those days of Teams, you know.
I guess while Christian is converting the file, I just jump in to make a quick reminder in case of your interest in Ricardo. Also shared a few weeks ago information in a postdoc position at the University of Costa Rica for isotopes fieldwork, in case you might be interested.
So some people is saying that maybe you have way too many apps open. People have seen some issues with using Teams and many apps open. So maybe that's another possibility of why we have some slow connection.
Closing everything down.
All right.
Yeah, it's exporting. So the thing is, something's really slowing me down.
Okay. Bueno, tal vez lo puedo decir en español ahora. Para todas las personas que se están reuniendo o están llegando al seminario, hay un archivo de Excel compartido en el chat que tiene dos objetivos. Uno es poder obtener información de contacto para poder tenerlos en una lista de correo y simplificar la distribución de los seminarios y la información. Y el segundo objetivo es tratar de ver el interés de personas en participar en una edición especial que estamos tratando de promover para la región de Latinoamérica, el Caribe. Así que si tienen gusto de ingresar al Excel, es un OneDrive. En el caso de que les pida una autorización, si fuese el caso, generalmente me llega el correo, yo lo acepto y pueden llenarlo en línea. Gracias. Vamos a esperar a ver qué pasa. Okay.
Okay, let me know what you see, please.
So we are seeing the first. We are seeing first slide. Yeah.
Second slide.
Yes.
Yes.
It's working now.
Okay. Sorry about that.
It's okay.
My sincere apologies.
There you go. There you go. Now you're fast.
Okay, let's give this a try from slide 3 then.
Yeah, and you can summarize it for the sake of the time as well. So go ahead.
Okay. Yeah. Okay. So we're still at the crossroads of why isotope-enabled hydrological models have not been picked up by the wider atmospheric sciences hydrological community as much as we and I would hope. There is potential. There are limitations, obviously. With this slide here, I kind of want to explain why models in general can be useful for management purposes, maybe moving beyond the academic applications such as learning about systems. So we think that models actually can inform decision making, that they can provide simulations and data, which would enable us to look a little bit into the future in terms of maybe scenarios, projections, etc. So there is this potential, but obviously this depends on model uncertainties. So how robust are our results and simulation? How much can we trust those simulations? Because we have scale issues in models. So it really depends very much at which scale you apply a model. Then there are the inherent model errors, starting with numerical errors. We have to simplify a complex real-world system into a model. And then there are parameter errors. There is the data issue as well, because your model is only as good as the data you feed it with. So there are those kind of issues, and I'm trying to show some workarounds and to get you at the where the potential for this data is. So why would we want to use isotopes in models to start with? Because what especially in hydrology, most people look at is the rainfall runoff transformation. So how is rainfall transformed into a hydrograph? And this is what you see in this slide here. But there are many other questions which are very relevant for water management. For example, where the water is coming from. So the water sources aspect is important. It is a huge difference if the water is generated from the soil or from groundwater, if water is stored in vegetation or directly returned to the atmosphere. So to get an insight on where the water is generated from may be hugely helpful to inform management. However, this you won't necessarily see from a hydrograph. So this is where the isotopes are coming in. They give additional information, a different point of view on system characteristics we can't immediately make available from our basic hydrometric measurements. There is another issue that kind of links the water quantity to the water quality through the concept of water age and transit times. So it makes a difference if water resides for very long periods in the system, or if it's more of a flow-through system and water gets very quickly delivered to the stream. So for water quality aspects, this is very, very relevant. And then there's also this ecological dimension, and there are applications which are informed by isotopes, such as, for example, stream metabolism, looking into processes of respiration and photosynthetic production. But there's also the water partitioning, the eco-hydrological water partitioning aspect of how much water is transpired by vegetation, how much water is lost to the atmosphere through canopy or soil evaporation. And I will be talking about this aspect of the eco-hydrology in isotope-enabled models in a second. So let me move along to the first, in that case, a plot scale application located in Berlin. So it's not a very tropical climate. It's actually almost semi-arid. It doesn't rain more than 400 to 500 millimeters a year. Berlin is quite a big city. It's very green, and pretty much every tree in the whole city is monitored due to issues such as the urban heat island effect, water uptake by those trees, and how much groundwater recharge they allow, because that groundwater is locally sourced for drinking water. So I will be reporting from an experiment that used in situ high-resolution isotope monitoring. So they measured a couple of different trees, and I will be focusing on a mature, almost 100-year-old maple tree, which is the most common street tree in Berlin. So just to link this up, from the plot scale to this wider management picture. I mean, we can talk later about the experimental setup, the instruments they used, how they really in the field monitored for in situ stable isotope data. But what I want to show is, you know, that there are differences and challenges, and there is quite a different information content in that data as well. So why do we want to do this in the first place is because there are many uncertainties related to eco-hydrological water partitioning. So there's always the question of how can we split the actual evapotranspiration into its components, into its hydrological processes. So this is one aspect where the isotopes come in handy as an additional source of data that allows us to determine at which depth from the soil plants take up water. So there we want to reduce those uncertainties and really pin down those fractions of transpiration, canopy evaporation or soil evaporation, and the depth from where the water is sourced, in that case for this maple tree I'm showing here in this picture. And almost the leftover then is, especially in this semi-arid climate, is then the groundwater recharge, which is important for the drinking water. So you kind of have to balance out, you know, planting trees might help with reducing the urban heat island effect, but it also might decrease groundwater recharge, which is very much needed for drinking water. So here I'm showing normalized xylem and normalized atmospheric vapor isotope data to make them comparable. What you see is a time series of 3 hourly isotope measurements in situ from beginning of April until the end of October. So this is a full growing season in the temperate northern latitude climate. And what immediately strikes is that there are quite some marked differences between deuterium and oxygen 18. So oxygen 18 is the darker color, green for xylem.
Cristian, creo que estamos en el
Yes.
slide, estamos todavía en el slide de el diseño experimental en el mapa.
It didn't move. Oh yeah.
It didn't move this time.
Okay, I'm switching back and hopefully it comes up. I don't know what it is.
Yeah, this is my first time seeing this.
PDF is not very heavy. Internet is okay. Nothing. Did it come up? Do you see dots? Data? Time
No,
series?
not the data yet. We are seeing the setup of where the location of the instrument was and the rainfall and the rain sampler and soil moisture and all of that. So we are seeing the aerial photo of the park.
Yeah, this is the previous slide.
Yes.
Damn, I'm going silent again. Trying to figure out what the issue is. Still nothing?
No, it's not moving. It's again stuck in that slide.
So maybe it's coming back up again. I just dropped out of the sharing mode. Can you see this slide ways forward?
Yeah, I think we're seeing the xylem and we're seeing the vapor isotopes now.
Okay. So now you see a lot of data, a lot of points.
Yeah.
That's the purpose of this slide. So one way to maybe move forward, reduce uncertainties, more robustly partition water could come through in situ isotope measurements. And what you are seeing in those time series here is 3 hourly data over a full growing season from beginning of April until end of October. The data is normalized to make the two isotopes comparable. The darker color is oxygen 18. The lighter green or bluish color is the deuterium. And what you immediately see is there is a lot of variability in the time series in general, and then there is a difference, at times stronger or more moderate between deuterium and oxygen 18, especially for the xylem data. So if you plot a moving average through the data of about 3 weeks, then you kind of see some patterns. And what we have here is that the first half of the growing season was much drier compared to August to October. So in terms of the xylem data, the difference between oxygen 18 and deuterium becomes more disparate. And for the atmospheric vapor data, this kind of comes closer together compared to the drier season. So we kind of see that there is information content, but there's also maybe a lot of noise we can't really explain yet, especially if it's biologically mediated processes, such as with the xylem isotope data. I will be switching slide now, so please let me know if what you see is a model scheme.
It is. Go ahead.
Okay, so the idea now is to use that data and test it out with quite simple conceptual isotope-enabled hydrological models. And if that data helps us to constrain those eco-hydrological fluxes, to make those data more robust, to trust our simulations even more. So I'm comparing two different model setups. To your right, it's the most simple model ever, two soil storages with a canopy interception storage. And then you have different parameterization, but you can, together with the isotopic mass balance, then calculate what is transpiration, what is canopy evaporation, and what is soil evaporation. This then is compared against a slightly more complex model, which discretizes the soil into 3 different soil storages: a very superficial soil horizon, then down to 30 centimeters, and then a third soil horizon down to 1 meter depth. And we also implemented different hypotheses into that slightly more complex model. One hypothesis is that the maple tree has access to the saturated zone, so deeper groundwater storage with almost unlimited water. So whenever the soil is dry, the tree might tap into that saturated zone water. Then there is also another hypothesis related to in-tree processing of water and isotopes. So we implemented tree water storage to test hypotheses if fractionation in the tree might affect what is ending up in the atmospheric vapor, or if the tree is just transpiring maybe fractionated soil water, but the transpiration process itself is non-fractionating. So those were kind of the model setups, playing around with different hypotheses to use those in situ high resolution data and see how far we can get with a still relatively simple model. So I'm switching slides. Maybe, hopefully, you see observed versus simulated time series. So if I'm not hearing anything, I assume this is what you see. So it's a multi-panel plot on top, rainfall over that growing season. It's hourly simulations and observed data. So the second panel is measured soil isotopes. The plot in between then is measured soil water, but of the deepest horizon down to 1 meter. And then you have simulations of the observed xylem isotopes for deuterium and oxygen-18. And the bottom panel is where the simulated maple tree is sourcing its water. So it's the fraction of the deeper soil storage that is used for the total transpiration. So what you can see essentially is that the simulated soil isotopes are pretty close to observations. It's very stable anyways, but the xylem isotopes are a lot more variable than we are able to simulate. So we kind of get those fluctuations, especially over the drier first couple of months into the growing season. So the model simulates fractionated water uptake from the soil, and then this big pulse of rain in August kind of resets the water isotopic signature in the tree, but it remains fairly stable, just depletes a little bit towards the end of the rainy season. So there is the thing that we can't fully represent the whole spectrum of observed xylem isotope variability. This then results in quite different, if we compare the two model setups, in quite different root water uptake depth. The simple model sources almost all of it. It's in water from the deeper horizon, while the slightly more complex model that discretizes the soil into more horizons or different soil layers kind of makes a bigger difference. But on average, it's about half of the transpiration sourced from 50 to 100 centimeters. So how do the atmospheric vapor simulations look like? So what we have here is plotted vapor LC excess. The blue dots are observations, and the lines, the black line is the more complex model. The gray line is the simple model.
Sorry, Christian. I think we're still seeing the previous. Okay, there you go.
It's a delay. I don't know what this is, to be honest. So it's delaying the switching of my slides. Apologies for that. So what you see here is then the simulated atmospheric water vapor isotope signature compared to the measurements, the in situ high resolution measurements. Again, there's more variability in the observations than we are able to capture with a simple model, but the dynamics and this almost linear positive trend in LC excess is fairly captured by both models. The more complex model does a better job in representing the peaks. So what we can gain from this is that the atmospheric vapor is a mixture of transpired water, it's a mixture of evaporated water, and it's also a mixture. So one component is locally transported moisture. This is something we all see together in this isotopic measurements of atmospheric vapor. To plot this as a bivariate plot, oxygen-18 versus deuterium, then you kind of see that the simulations fall pretty close to the observations, but again, they can't really represent the complete spectrum of the variability, with a slightly better performance of the more complex model. I will switch slide to the eco-hydrological water partitioning. So those are the simulations, median simulations of the model for the drier first couple of months and the wetter last couple of months of the growing season. So I hope you see this slide. If not, let me know.
Yes.
Okay, so it came up. You have the simulated water balance there. It's essentially the same for both models, but the difference is in partitioned soil evaporation. So there, the more complex model does a better job because it fully takes into account the fractionation, the evaporative fractionation that is happening in the first upper 10 centimeters of the soil layer. So there is a difference in terms of model outcomes. And then if we kind of plot the simulated vapor according to the transpiration to AET fraction, then we temporarily see when most of the atmospheric vapor consists in transpired water, or if the soil or canopy evaporation becomes more important. So evaporation is the coffee brownish color, and the greener it gets, the more bright the green, the higher the transpired fraction of total AET is. So in the beginning, when it's dry at the start of the growing season, this is something we would expect. So the tree starts to grow and transpire. So close to 100% of the total actual evapotranspiration is transpired water. But then there are those spikes, those peaks of evaporation kicking in, especially in drier conditions. So when the atmosphere is relatively dry, then most of this vapor is a mixture of evaporation sources. So this is information we can get out of atmospheric vapor data. And I will be switching and moving up in scale, so away from the high resolution in situ modeling to the more conventional water sampling with automatic water samplers, and bringing back bottles into the lab to analyze them for isotope. This is still what probably 99% of the research group and institutions are doing. The management issues in Costa Rica here specifically are land use transformation in that catchment, where we have worked quite a lot. So pineapple monocultures were cropping up over the last 20, 30 years, transforming quite large scale landscapes from pastures and forests into monocultures.
Did you move the slides?
Yes, I did. Damn.
So we're still seeing the transpiration slide.
Okay, let's give it a second. Maybe it's still just the delay. Still not there.
Not yet.
Damn it.
It's all right.
I'm just dropping out of the presenter mode, bringing it back up again.
Yeah.
Are you seeing a map now?
Yeah.
All right, of northern Costa Rica. So we're moving away from the in situ monitoring towards to the more conventional water sample monitoring, bringing back samples into the lab for stable isotope analysis. And the management issues here are in northern Costa Rica are land use transformations that pressurize the resources. And obviously there is a pretty dynamic climate variability in here. So we have drought periods that kind of interact and interscale and overlap with flooding. So what we did is we instrumented in the southern corner of this large scale 2,500 square kilometer catchment, a headwater catchment, which is almost pristine. So rainforest, no human intervention there. This was our test bed for models that then we scaled up to simulate the whole 2,500 square kilometer catchment, hopefully getting some answers about how the land use transformation affect hydrological processes such as transpiration and evaporation. So I will switch slide to show the headwater catchment monitoring that took place over a couple of years from 2013 to 2019. I'm talking about 3.2 square kilometers. Do you see the slide? I hope so.
It is okay. Go ahead.
Okay. So we kind of intensively monitored that catchment for its tributaries at the catchment outlet with isotope monitoring in rainfall in the tributaries in the main river. We monitor it for soil moisture, for the meteorological variables, to come up with a water balance, an initial water balance. So it rains quite a bit, only has a dry season of 3 months, but it still rains. So we're talking about close to 3 meters of rainfall a year, with surprisingly low actual evapotranspiration rates of only 0.6 meters per year. So this is the headwaters. There is a lot of cloud cover for most of the year, so it's rather energy limited. And this explains partly that stream flow is pretty high with 2.2 meters per year. So now we were setting up an isotope-enabled model trying to explain certain differences in forest structure, which we observed, because if you look at that inlet map here of leaf area index, so the southern slope showed lower leaf area index consistently compared to the northern slope. So this was something we wanted to test in a isotope-enabled hydrological model. So I'm switching slides, and what you should see is a conceptual model setup of a spatially distributed isotope-enabled model we've been extensively using in Costa Rica and abroad. So here we specifically accommodate the interception of the rainforest. We incorporate the isotopes into the water balance, trying to separate out or partition the incoming water into net precipitation, transpiration, and then the different evaporation components. And then what the model allows you to do is not only can you simulate the hydrograph or discharge observations, you can also use the isotopic information recollected in the stream to calibrate the model, and that then allows you to estimate transit times and water ages, and gets you a step closer to look into water quality. So how looked the results for this catchment? We arbitrarily singled out 2 points, P1 on the southern slope, situated or located right in an area of low leaf area index, compared to point P2 you see on the top panel. So this is simulated transpiration compared to in blue precipitation rates. And what we found with the model is that the geomorphology and the catchment structure influences the forest structure, and then also finally the transpiration rates. So the lower the vegetation density, this is mostly situated on the higher slopes, less soil development, and then this also resulted in significantly lower transpiration rates. So this is something we could test with this isotope-enabled hydrological models, apart from looking into transit times and water ages. So there are, together with the transpiration rates, there are differences in the age of that transpired water. So the southern slope with less transpiration rates processes the water more quickly. So transit times are faster compared to the more developed lower slope and higher transpiration in the northern part of this catchment. So this is something you can look at. Management issues here it's a part of a biological reserve. So this is the most stringent protection category in Costa Rica, sits even above a national park. So here you can't do anything, no interventions whatsoever. But always the question pops up of how relevant is this protection scheme? Would it not just be better to allow some type of management and maybe take out some wood, whatever, for commercial purposes? So this is then something you could test with this model as a scenario to see how that modifies the eco-hydrological water partitioning. However, we still face the issue that we are looking at a very small scale, and the whole catchment is much larger, and probably societally more important, more relevant management questions need to be asked at this 2,500 square kilometer scale. So what do we do in that case? Because we don't have information everywhere, so we need to do something. And our approach in that case was to substitute time for space. So instead of developing intense or pretty high resolution time series at one point, for example, the catchment outlet, we did a monthly sampling of 46 streams in that catchment, trying to develop a spatial picture, a spatial isotopic picture, which we then can use to calibrate and evaluate the upscaled model. So what you see here is type of data we need. We need to differentiate land use. In that case, the model uses topography to direct drought water towards the lowest downstream point. We need leaf area index. And most pressing is the issue of input data, such as your meteorological variables, and with that come the precipitation isotope, where we had measurements in the catchment using 3 stations of a national isotope network developed by Ricardo. But if you interpolate that, then you kind of get very coarse interpolations. So one idea then was actually to use isotope-enabled climate models that potentially can provide input you can feed into hydrological model. So working with atmospheric sciences, we used meteorological input and stable isotopes in precipitation from a regional isotope-enabled climate model, which is called ISO-RSM, to feed and run the hydrological star tropics model with. So we should essentially get a spatial picture of our inputs, a better, more complete picture of those spatial variabilities in the input, which then should positively affect our simulations. However, you can't just directly take climate model output and put it into a hydrological model, because obviously there will be a bias. There is some type of correction needed, and this is what you see in this slide here. So to make this data hopefully more useful for the hydrological model application, then we kind of performed a type of bias correction. So for those 3 stations, you see in red measured data, and then the lines are bias corrected climate model output for that pixel where the measurement station is located. You immediately see that there are challenges. So the extremes you measure are not represented by the climate model, and even the bias correction does not quite get us there. But you almost perfectly represent the seasonality and the in turn intra-annual variability of your system. So this seems definitely well worth exploring in a hydrological model application. This is what we did. So we fed this data, the bias corrected climate model isotope data, into the hydrological model to run simulations. And this is what you observe in this next slide. So for different stream flow gauging stations, we had bits and pieces of data available. You kind of see what the model does in terms of different performance criteria. It's not perfect, but it's actually quite decent if you think of that the input data is coming from another model adjusted, but it's still another modeled source of data. So the model represents the hydrograph peaks, the troughs and the lows, and then even the isotope simulations at some selected points where we had this monthly sampling available are actually fairly well represented. So we thought this is an encouraging result, and this should enable us to look into how much is transpired by different vegetation covers, etc. So the next result I'm showing here is different land cover management scenarios of the model as maps, which is something that is probably more easily communicated to water managers if you get around explaining what transpiration and actual evapotranspiration is. So if you look at those maps down there, you get a spatially distributed picture of annual transpiration rates, and then you can tease out that how much is transpired for this area where the pineapple monoculture sits, how much do the forests, the protected forests transpire versus grasslands and other crops. So with that and the additional isotope data, we were actually able to constrain the water balance estimates at a minimum on a monthly and most definitely on an annual basis. So there the isotope data really offers additional information to calibrate and evaluate your model and constrain the simulation towards more robust results. We also used the same approach now in Ecuador, and I'm just showing some initial results from an application in South Africa with another spatially distributed isotope-enabled model. The IEA is using and promoting. So this is not competition between our model and what the IEA does. This is just another brilliant effort to promoting a more widespread use of this type of models. So they simulated a larger semi-arid catchment with a very seasonal climate using a model called J2000, which is isotope enabled in a very similar way to my group's model. But then what they try to tease out is, okay, so you can use your measured data, but you have the difficulty of only a couple of measurement stations. You need to interpolate over a large area. You could use a regional climate model, which is computationally more intense, but you could also directly use a global isotope-enabled climate model, such as the ISO GSM, to run and produce hydrological simulations. So they tested out which isotope input source performs better in simulating eco-hydrological simulations. Those are the comparisons. See is that the regional climate model produces better fits to observe data compared to the global model. The global model underestimates if it's not calibrated or corrected, particularly underestimates with a pretty consistent offset throughout the time series of your isotope input. So that clearly shows that you need some type of correction, but it still is able to match measured groundwater isotope signatures if used in simulation with a hydrological model. So even that is encouraging for a more widespread use. And this also then allows you to look into different hydrological processes, such as the actual evapotranspiration per year, how much of your soil is saturated and where and how much of groundwater recharge you simulate based on the isotopic simulations in that type of hydrological model. There is variability. The spatial patterns are quite similar, which is encouraging. And then you kind of face the issue that you need to tease out. So if you settle on, for example, the regional isotope-enabled climate model, how much do you trust those simulations? So this needs to be compared, evaluated against some other external measurements, such as groundwater measurements in wells, such as stream flow isotope measurements. So even a couple of points, a couple of data measurements help in that regard, constraining the model towards more consistent simulations. So with that, I would just want to cut here, quickly draw some conclusions, and maybe hopefully even with a massive delay, there are some questions. So I'm sorry about this. So I hope that I kind of showed you that there is potential in using isotopes in hydrological models. You can use isotopes to develop a model because it helps conceptualize processes, but then you also immediately have additional information to calibrate and evaluate your hydrological model simulations, which produces most of the time, in our experience, more robust, more constrained simulations. The other point is that with the isotopes, additionally to hydrometric measurements, you can get at partitioning eco-hydrological processes. So you can tease out how much is transpiration, how much is evaporation. So you are internally consistent and can look at, for example, land use scenarios, land cover transformations. And then I think there is huge unrealized potential in coupling hydrological model to climate models, which are also isotope enabled. And that kind of bridges a little bit of the gap between the isotope hydrology community and the isotope atmospheric sciences. So with that, I thank you very much for your attention, and maybe there are still questions if you're there. I don't see anybody anymore.
Thank you, Christian. I think I'm going to switch to Spanish because I think we have a very large community from Latin America. So, muchas gracias, Christian. ¿Alguna pregunta de la audiencia? Hay muchas personas que no conozco, así que por favor adelante si tienen alguna consulta. Esto es un espacio totalmente amigable para hacer las consultas que quieran al respecto. ¿Nadie?
Tal vez en el chat.
En el chat no vi ninguna pregunta todavía, pero yo te puedo hacer una pregunta, Cristian.
Adelante, por supuesto.
Sí, en este momento existe un gran debate en términos de qué tipo de tecnología o metodología debemos utilizar para medir isótopos en el suelo y en las plantas. ¿Crees que sea posible en este momento, porque es algo que yo he estado pensando, tratar de poder colectar muestras, analizarlas bajo diferentes técnicas e introducir esos compuestos, esas composiciones isotópicas dentro de los modelos para ver qué, es decir, para ver si le ingreso al modelo composición isotópica de centrifugación, qué resultados obtengo, si le pongo al modelo vapor medido directamente, qué resultados obtengo, si lo que hago es tomar muestras y me las llevo al laboratorio para analizar el vapor en el laboratorio, o si son muestras de equilibrio, o finalmente si son muestras de extracción criogénica. ¿Alguna idea al respecto en ese sentido? Y lo otro es que tal vez alguna consideración tuya al respecto en términos de que estamos tratando de movernos hacia la instrumentación y la medición in situ, pero no son esfuerzos que se puedan sostener en el tiempo. Sí, podemos medir una época de crecimiento y luego tenemos que sacar todo el equipo del campo y solo medimos una época de primavera y de verano y el otro año podría ser muy diferente hidrológicamente y las cosas podrían ser muy distintas. Así que yo veo como eso está muy bien, estamos conociendo algo, pero como que al mismo tiempo estamos generando vacíos porque estamos tratando de cortar el monitoreo continuo tradicional para técnicas en campo cuando no se pueden sostener en el tiempo. Eso son dos cosas distintas. Una tal vez una opinión y la segunda es de modelaje.
Perfecto. No, Ricardo, muchas gracias. Hay un montón de debate acerca de los dos puntos. Tal vez voy a empezar con la última parte relacionada con el monitoreo in situ, ¿verdad? Y esos datos de una resolución, una frecuencia temporal muy alta. Entonces, eso es valioso, un esfuerzo que vale la pena porque se generan datos, ¿verdad? Con contenido de información que antes no hemos visto. Pero ahí está el problema porque llega a un costo muy alto. Eso no es instrumentación que puedes dejar ahí, ¿verdad? In situ operando ni siquiera un par de días. ahí necesitas a alguien, ¿verdad? Que revisa, que calibra diariamente tu instrumentación. Entonces, eso es tremendamente costoso y no se va a poder replicar en muchas partes del mundo, ¿verdad? Todavía en Costa Rica, por ejemplo, yo veo eso difícil. Em, y con ahí nada más tuve esa experiencia ahora, ¿verdad? Trabajando con un grupo en Alemania donde lo hicieron, donde hay los recursos, ¿verdad? Entonces, donde sí produjeron una serie de tiempo casi que consistente, sin huecos, ¿verdad? Porque tenían el personal ahí en campo, sentado a la par, ¿verdad? Del picarro, ¿verdad?
Eh,
Acampando ahí,
eh,
bajo el árbol, literalmente. Entonces, ahí sí fue posible, pero usando esos datos también nos dimos cuenta que no estamos al 100 seguro qué estamos viendo,
yeah,
¿verdad? Porque ahí puede haber muchos procesos que ahora actualmente todavía no tenemos mucho conocimiento. Sobre todo ahí hay influencia biológica que no sabemos en su totalidad, ¿verdad? Cómo impacta, cómo influencia, cómo modifica em, esa composición isotópica. Entonces, es un dato útil, ¿verdad? Para anclar tu modelo, pero también hay muchas desconocidas todavía.
okay,
Esto, y eso es mi percepción ahora, un dato más factible, más estable y tal vez más reproducible, ¿verdad? En diferentes sitios a una escala mayor es vapor. Entonces ahí, ¿verdad? Nada más conectar un tubo al picado ¿verdad? Dejar colgando el tubo por la ventana del laboratorio, por ejemplo, ¿verdad? Eso ya te da un dato, es mucho más sencillo de medir y más estable. Y ese en términos del modelado para la calibración y evaluación ayudó más en producir simulaciones más robustas de transpiración y evaporación. Entonces, ahí veo mucho potencial.
okay,
Ahora, la otra parte, ¿verdad? Está un poco relacionado, porque hay un cierto sesgo según el método que utilizamos para analizar las muestras, ¿verdad? En plantas, en el suelo. Entonces, ahora en ese momento hay un montón de discusión, un poco el método establecido es la extracción criogénica. Pero también, ¿verdad? Si metemos esos datos en un modelo, ahí también hay artefactos y no sabemos exactamente, ¿verdad? Con qué tipo de agua estamos lidiando porque extrae todo. Y para efectos del modelo, ¿verdad? Lo que necesitamos es la parte móvil del agua. Entonces ahí tenemos también un sesgo, ¿verdad? Metodológico que para el modelo puede representar problemas. Entonces hay otros métodos, ¿verdad? Que para efectos del modelo ahí sí, centrifugación, ¿verdad? Métodos que enfocan en la parte móvil, extraíble de suelo, de plantas, ¿verdad? Entonces, eso para efectos del modelo es más útil de usar, porque sé con qué tipo de agua y proceso estoy trabajando.
okay. Ok, tenemos otra pregunta de Ana.
Bueno, muchas gracias, Cristian. Yo soy Ricardo también para la organización del seminario. Yo tenía una pregunta justamente sobre la parte del escalamiento, ¿verdad? Porque
Ujú.
las mediciones se hacen muchas veces incluso a nivel de individuo que no tiene la misma edad, el mismo tamaño, no es la misma especie que todo lo demás que puede estar en el área dentro de la grilla de resolución del modelo. ¿Qué podrías comentar un poco sobre la experiencia del trabajo de la partición ya haciendo el escalamiento, por ejemplo, con datos medidos en campo de evapotranspiración?
Sí, eso es un gran punto, ¿verdad? Y muchos de los esfuerzos enfocan en ese ajuste de escala, ¿verdad? Alejarnos un poco de la escala em, parcela o cuencas muy pequeñas, ¿verdad? A escalas relevantes para el manejo, ¿verdad? Cientos o miles de kilómetros cuadrados. Entonces, como no podemos medir en todo lado, como estamos usualmente, ¿verdad? Un poco limitados en dónde y a qué frecuencia temporal podemos medir. Entonces, para nosotros fue muy importante identificar que en parte podemos sustituir esas series de tiempo largas en un punto por una imagen espacial. Entonces, al final, ¿verdad? Y me refiero directamente al estudio en el norte de Costa Rica, en esa cuenca más grande. Entonces, fue igualmente de valioso monitorear mensualmente en muchos ríos, comparado con una serie de tiempo, ¿verdad? Diaria de 3 años en la cuenca arriba. Entonces, hay ciertas alternativas que ayudan, ¿verdad? De llevar el muestreo para que todavía nos dé información útil, por ejemplo, para modelos que nos ajustan la escala hacia esa relevancia en el manejo, para el manejo.
Gracias, Cristian. ¿Alguna otra
Gracias.
pregunta? Enrico. Te escuchamos, Enrico. Creo que estás silenciado. No te podemos escuchar, Enrico. Tal vez puedes poner la pregunta en el chat. Ahí está. Dice que tiene uno. Vamos a ver si escribe la pregunta en el chat. Mientras Enrico nos pasa la pregunta, no sé si existe alguna otra persona con alguna consulta. Si la puedes escribir en el chat, Enrico, sería genial.
Otra persona con problemas en Teams.
Sí, fue problemático ya. No pasa nada.
Teams es problemático.
Muy temperamental. Hoy estuve con 3 plataformas diferentes.
Un poco para saber cómo controlan problemas de condensación en los espectroscopios, en los monitoreos continuos de vapor. Bueno, eso es una muy buena pregunta. Hay no solamente problemas de condensación, hay problemas de, hay que tener cuidado con la limpieza de las líneas, hay que estar tirando a aire seco, limpiando líneas, hay que hacer, hay periodos de estabilización, hay que controlar muy bien el flujo que entra al equipo porque eso puede dañar los equipos. Así que es un tema bastante complicado. Cuando funcionan bien, pero hay que hacer intercambio entre el vapor de la muestra y el estándar, porque hay que estar calibrando el equipo de manera constante. En algunos casos hay que controlar temperatura. Así que mi pregunta iba por ese sentido, porque yo pienso en la región. Bueno, el seminario es para la región de Latinoamérica y el Caribe. Estamos muy lejos de eso, de tener equipos monitoreando, por decir algo, en la parte alta de los Andes continuamente. Así que yo creo que tenemos que, y tal vez el seminario y la iniciativa se pueda prestar como para un paper de opinión de estrategias de monitoreo viables para regiones como Latinoamérica y el Caribe, donde no tenemos todavía ese tipo de presupuesto y se podrían, por ejemplo, el ejemplo que presentó Cristian sobre monitoreo espacial en un año hidrológico en casi 40 sitios, puede ser una iniciativa totalmente viable para grandes cuencas y que puede producir muchísima información espacial. Dice aquí, existe buena aplicación para esas preocupaciones. Sí, dejar los espectroscopios encendidos, digamos, no sé si te refieres en el laboratorio y en la época del monzón. En lo que es el laboratorio, pues lo que se recomienda siempre es tener fuentes de poder que puedan permitirte continuar utilizando el equipo y evitar cortes de corriente, si estás hablando en el laboratorio. Y ninguna torre te va a permitir de suplemento de energía mantenerlo tal vez 1 o 2 horas para poder terminar la corrida si el corte de electricidad va a ser largo. En campo es exactamente lo mismo. Tienes que tener una buena fuente de poder para poder garantizar de que no van a haber cortos de corriente. Esos equipos generalmente están construidos para trabajar con lo que se llama energía sucia, así que soportan entradas fuertes de corriente, etcétera, pero eso puede producir problemas. No tanto cuando se analiza vapor, porque no hay jeringas. Cuando hay jeringas y es agua líquida, todos los problemas eléctricos que puedan ocurrir terminan siendo un desastre para el equipo en términos de utilización de las jeringas y las inyecciones. Pero en fin, en general, creo que sí estamos un poco largo de poder tener esas mediciones continuas. Sí. Ok, bueno, creo que no sé si hay alguna otra pregunta. Nos hemos extendido 25 minutos de lo esperado, pero fueron por esos problemas de comunicación que vamos a ver si los podemos evitar para la próxima. El próximo seminario va a ser en 2 meses. Yo enviaré nuevamente anuncios y información sobre la persona que nos va a dar el seminario, etcétera. Y recuerden que tenemos como una semana más para tratar de enviar los resúmenes preliminares de algunas ideas que tengan para un evento de una edición especial. Y posiblemente haya que extenderlo, pero recuerden que si necesitan información, nada más me envían un correo y yo les puedo enviar la información. Así que vamos a dar por concluida la charla de hoy. Cristian, de nuevo, muchas gracias por el tiempo, muchas gracias por aceptar ser el primero en lanzarse al vacío latinoamericano y del Caribe en esta iniciativa, pero creo que esto va a ser el inicio de muchas colaboraciones futuras. Y si necesitan el contacto de Cristian, si no lo tienen, me pueden escribir y yo con mucho gusto les paso el correo para que puedan comunicarse con él directamente. Bueno, que tengan buena tarde y luego les paso el link del video que hemos grabado.
Perfecto, muchas gracias. Excelente iniciativa. Estamos en contacto.
Hasta luego, bye.